Thank you for that intro, and thank you very much to our hosts for putting this lovely talk together and giving me the opportunity to get up here. I would say that they saved the best for last, but let's be real. I'm going to be talking about colorectal cancer, so they wanted to make sure that the poop jokes were as far away from lunch as possible. Either that, or they wanted to make sure everyone could get to a good stiff drink as soon as possible once I stop rambling. One of those, maybe both. All right, I guess I need to figure out how this thing works. Big green button, send me to school. Okay, so I'll start off with a little bit of a intro to New Day Diagnostics, and then I'll go into our development project, and then leave you with some of our next steps. So I was originally with EDP Biotech, early detection products, and then we've recently merged with New Day Diagnostics because we share a clear vision to improve lives by empowering people with innovative and accessible diagnostic solutions. So prior to COVID, EDP was focused on our Coloplex assay, and we had just finished a large study at the end of 2019. We were gearing up with a Series A fundraise. We were getting ready to commercialize our product in the European market, and COVID hit. And that, I wouldn't wish COVID on the world. At the time, it was a huge roadblock for us, but it actually ended up letting us do some of the things that we had wanted to do for a long time. My CEO had wanted to start up a CRO business within our business to offer our services, and he had also wanted to set up a clinical lab to get ready to test our products, and that's actually how we met New Day. We ended up helping them with their EUA study, and with our time in the trenches with them, our companies really gelled and worked together, and we were able to bring both companies together into a soup to nuts diagnostic company. So New Day brought a product pipeline and additional resources to the new company. Another really exciting thing we were able to do was to bring on a mergers and acquisitions department full time. Sam Libby has been instrumental, him and his team of TCB Capital. I'll mention a little bit more about that in a minute. Our company has kind of three main areas that we focus in. New Day has brought a product pipeline, and in addition to the pre-existing CRO services, so we basically had a startup within a startup doing CRO work for initially EUA studies, everything from serology to molecular, to lots of rapid antigen studies, doing both the analytical validations and the clinical performance evaluation. So we are able to take those capabilities and combine them with the product pipeline from New Day, and really there's a lot of synergies there that we're able to capitalize on. But our main attribute is our diagnostic laboratory. So we are ISO 13485 medical device certified, and having that research component was what led to the kind of organic startup of our CRO services. And now we also have a CLIA laboratory offering diagnostic services, originally focused on serology, on antibody testing for COVID, and then we picked up the testing for our local health department, diagnostic testing for SARS-CoV-2, and now we're excited to beginning of next year launch our colorectal asset out of our own clinical lab. So the existing foundation of EDP plus New Day is what really brought the two companies together. And I want to move on to colorectal cancer screening. Colorectal cancer is still the third leading cancer for both men and women in terms of incidence and mortality. So it's a very important disease, very prevalent worldwide. There are a lot of the existing screening modality in Europe is FIT testing, so fecal occult blood testing. Whereas in the U.S., we are primarily focused on the colonoscopies on the 10-year plan. But there's a large problem, especially here in the U.S., with both compliance to fecal occult blood testing and to compliance with colonoscopies. And the important thing with colorectal cancer is early detection. That's why we've always been focused on early stage 1, 2 colorectal and medium risk and up polyp detection. And there's a huge opportunity for blood-based diagnostics for colorectal cancer. That's the holy grail. That's what everyone is currently chasing. And we're focused on screening, triage, and surveillance for colorectal cancer. The aims of the study that I'll be going over were to validate a biomarker set and algorithms that we had picked based on a sample set that I'll show you in a minute that was primarily early stage 1, 2 colorectal and polyps. We're focused on reducing the endoscopy burden because of these large screening programs in Europe where they're doing population-based screening for colorectal cancer using fecal occult blood testing. They have more people that need colonoscopies than they have doctors to perform those colonoscopies. And various countries are responding responding to that endoscopy burden by doing things like changing the FIT cutoff. And I'll show a slide in a minute that specifically goes into the repercussions of that approach. We're also focused on providing a alternative method of testing for individuals that can't take a FIT test because they have pre-existing conditions that involve bleeding. So when we started out to build our multiplex assay, we started with my main scientific advisor, Dr. Herb Fritchie. He was the chief of clinical chemistry at MD Anderson for 42 years and has a tremendous amount of experience moving cancer diagnostics through the FDA. He and I sat down and we did an extensive literature review and spent a lot of time going through our internal data. We had performed various mass spec immunoprecipitation experiments using normal versus tumor tissue and normal versus colorectal cancer serum samples. And we had a list of kind of the known pathways that we wanted to focus on. And the Luminex xMAP platform was just a really great way for us to screen those initial targets. We started out with 38 biomarkers that we were interested in, and actually it was Millipore Sigma that first turned us on to the xMAP platform. They one of their guys, it was Sean Connor at the time, came by, showed us this mag pics, placed one in our lab for a couple months as a demo, and we were hooked. So we ran 8 different multiplexes looking at 38 biomarkers on actually this sample set that I'll go over briefly here. It was samples from a collaboration with the Vidor Hospital over in Denmark. We started focused on clean colonoscopy versus early stage 1 and 2 colorectal, and then we added in low-risk adenomas and high-risk adenomas for our initial biomarker discovery phase, focused on sorting out medium-risk adenomas and up from low-risk adenomas and clean colonoscopies. And Vidor Hospital was a really great collaborator. Hans Nielsen was very, very gracious to provide those samples, and we focused on multiple different machine learning modalities in order to come up with the biomarker combination that we then put together for a single multiplex assay on the xMAP platform. With a kind of a wide array of markers, most of which are some of the usual suspects, but there are some more oddball ones out there, like that keratin 1-2 complex. The mass spec guys thought I was Looney Tunes when I first keyed in on that one, because it's the most common contaminant in any mass spec, but it showed up again and again, and it actually ended up being one of the ones that performed rather well in the assay itself. So next, we went on to a larger sample set of nearly 2,000 samples that we split into a training and a test set. The training and this sample set is heavily weighted towards early stage colorectal and polyps, with about 56% or 54% of each of one of those sample sets being low-risk adenomas and clean colonoscopies. And I guess another thing to point out about this sample set is this is a FIT positive sample set. So this is all downstream of that positive FIT test. So all of these individuals would have gone on to a confirmatory colonoscopy. Or I should say they did, but in the hypothetical screening setup that we have here. So this is linear regression, looking at each of the biomarkers. One of the things that we did before sitting down and running all of these samples that I'm really glad that we did, we made a pooled normal human serum controls of our own that we spiked with the calibrator material for all of these samples, and then we aliquoted those out, banked them in the freezer, and then every single run we pulled those off and we put fresh samples on so that we could look at run-to-run variation, plate-to-plate variation, day-to-day variation. And that was actually another one of the factors that went into, or that is going into the selection of which biomarkers are going to go into our final kit, because 16 biomarkers is great for research, but that's way too many for a clinical assay, especially to get meaningful information out of a machine learning algorithm. So the 10 that are highlighted there in green are the ones I'm going to show a couple more slides with those, but we've done other versions of this data where we've cut that down to 5, 6, and even as low as 3 samples. Because ultimately we'll want to reduce that to avoid some of the noise. But I had mentioned the plate-to-plate variation. That was another factor that let me select for the samples that, or the biomarkers that give the most consistent performance from plate to plate without some of the variation that you can get in these biological systems. So this figure here, this is highlighting IL-8. IL-8 was one of the markers that performed rather well, even by itself. Matthias from Vidor set this figure up. This is a violin plot, and the thing I'll point out here, so we've got clean colorectal, low-risk adenoma, medium and high risk adenoma, stage 1, 2, and stage 3, 4. And there's a very strong, significant difference between stage 1, 2 expression versus clean and low risk, which is exactly what we're going for with our biomarker selection process. And another thing that I really liked about the way we set this particular study up was we were, because of the collaboration with VDOR, we generate all the data, we put the assay together, but then we turned it over to them and let them do all of the analytics independent of us. That was also an approach that we took to multiple machine learning groups to look at the best way to build an algorithm with these markers and look at different approaches to pick the most efficient ones. We worked with experts in support vector machines, random forest, and imperial classifiers, to name a few. And it let us give them the training set with all of the answers, and they could build the algorithm as well as they could, and then we kept the holdout set and never divulged the actual the actual sample IDs for that, and we were able to keep it separate. This is an example of a support vector machine. This is a rock curve. It's not the most impressive curve ever, but what's really nice about it is the green line and the blue line are pretty much laid on top of each other. So we didn't have the breakdown that you can see quite often with overtrained algorithms, where the holdout test set just completely breaks. Especially with random forest, I had one colleague that would train up a random forest, and it would be a perfect, right angle, give them the test set, and it would just be a flat line. So this was really encouraging to see data that held up really well to that outside set. And just another point about this that's like, in order to understand this, you have to realize this is within a fit positive set. So this isn't the performance of the combined assay. The performance of the combined assay would be closer to 70% sensitivity, 96% specificity, because the goal here is to provide a triage assay to reduce the endoscopy burden while missing as few cancers as possible. And really with that focus of not missing the early stage and polyps. And within that set, we were able to maintain a high sensitivity and specificity to those early precancerous polyps and stage 1, 2 cancers. I like this figure quite a bit because it shows the real world application of that last rock curve that I just showed you. The first table here, this is 100 nanogram per ml fit cutoff. So this represents kind of the standard cutoff that was being used by most countries when FIT was first adopted as a screening program. That's what the Danish still use, and that's what was used for the 2,000 sample set that I just showed you. And then Matthias was able to model the effects of increasing the FIT cutoff and what that does to both the endoscopy burden and the number of cancers detected. So this blue line represents cancers that were detected. And 100% means 100% of the cancers that were detected by FIT at 100 nanogram per ml cutoff. And then here, the endoscopy savings is 0, because this is our baseline. And then this shows what happens if you raise the cutoff to 150 nanograms. You miss a few cancers, you save a little bit of your endoscopy burden. And then as you raise it higher and higher, you're missing more and more of your cancers, especially your early stage, in order to get a higher savings of unnecessary colonoscopies. But we were able to show with our data that using a blood-based biomarker assay as a triage or an adjunct to that FIT, we were able to reduce the endoscopy burden more than if the FIT cutoff had been raised to 150 while missing fewer cancers. So this is a good real-world example of how this strategy could be used to reduce the endoscopy burden, but while missing as few cancers as possible. and that's, this slide pretty much says what I just already covered. But kind of shifting gears slightly to the US. So the US doesn't have the endoscopy burden that a lot of European countries do. So there's not a need to reduce endoscopy burden. In the US, our focus is what can we do to increase colonoscopy compliance, to get the right people to colonoscopy, to make those colonoscopies more effective, to hand the GI doctor a tool that basically gives them a red flag, hey, slow down, spend more time on this patient, and you'll increase your adenoma detection rate. So it's kind of a opposite approach to a screening program from what some of the other companies have done. And so I mentioned that we also were able to bring on a merger and acquisition team. So right on the heels of our merger, which we completed about four months ago now, right after that, in fact, during that merger, we were also instituting an acquisition. So we purchased the assets of epigenomics, including a FDA-approved blood test for colorectal cancer and their IP portfolio. So as I move on to my last couple of slides talking about next steps, our next step is to take our existing xMAP protein assay and take a multi-omic approach by adding methylated septin-9 to that in a multi-omic test to boost sensitivity and specificity to the point where screening could be an option. And this is just very, very preliminary. I can't stress that enough. This is a small sample set, but it indicates that this approach could be effective and could get us back up to above that threshold we would need for CMS reimbursement. This is data on a 241 sample set, including 70 colorectal and 173 no incidence of disease detected. And using a multivariate logarithmic regression, we're able to show a 84% sensitivity with a 90% specificity. So like I said, very, very small sample set, so I'm not hanging my hat on it yet, but this is what we're busy at work, expanding this to a larger data set and seeing what, and also this is a very limited number of protein markers. This was only 3. We're now expanding that to the full 16-plex, and then we'll work down from there. But yeah, thank you very much for your attention, and I'd be happy to take any questions.